Hello children, welcome to biology class. Today we are going to discuss the lesson photosynthesis in higher plants. So this is the last lesson children. So as per the revised syllabus, this is our subtopics of this lesson. So the first two topics you are aware. Photosynthesis is a mean of autotrophic nutrition, site of photosynthesis, and the last one, factors affecting photosynthesis. That is your assignment topic. Then let's start with the first topic, pigments in green plants. So here the principal pigment is chlorophyll A. The necessary pigments are there, that is chlorophyll B, C, D, carotenoid and phycobionts. And you just study about this chlorophyll, which give green color, and the cyanin give red color, carotene orange color, xanthophyllus yellow in color. And this graph and explanation is there. Okay. So we know that light can be reflected, transmitted or observed by objects. A plant is green because it absorbs all color but green. Photosynthetic pigment absorbs lights for energy. Okay. Then about the light reaction. Children, what is light reaction? Or it is also known as photochemical phase. So in this picture, both light and dark reaction is exhibited. So this is light reaction, this is dark reaction. And in this photochemical phase, it includes the light absorption. See, this is the first phase, light absorption. Then splitting of water. Then oxygen release. Okay, and the formation of high energy chemical intermediates, that is ATP and NADPH. These much reactions are included in light reaction, and the formed ATP and NADPH are used in Calvin cycle, and it does not require light. That's why it is known as dark reaction, and the byproduct sugar is formed. So this is the complete steps of photosynthesis is exhibited here, but we can divide into the light reaction and dark reaction. So which are the steps included in light reaction? Yes, first one, the light absorption. This is the first reaction. Second reaction, splitting of water. Third reaction, release of oxygen. Then fourth one, release of intermediates. Okay, so the pigments are organized into two discrete photochemical light harvesting complex, LHC in short, within the photosystem 1 and photosystem 2. Children, this photosystem 1 and 2 named in the sequence on their discovery and not in the sequence in which they function during the light reaction, because while explaining it you will understood light photosystem 2 works first. Okay, that's why I gave this explanation. The light harvesting systems are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all the pigments, forming a light harvesting system also called antennae. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystem. Whereas in photosystem 1, the reaction center is chlorophyll A just have an absorption peak at 700 nanometer. That's why it is called P700. While photosystem 2, it has an absorption maxima at 680 nanometer. That's why it is called P680. Okay. Then the electron transport in photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into an orbit farther from the atomic nucleus. These electrons are picked up by an electron acceptor, which pass them to an electron transport system consisting of cytochromes. This movements of electrons is downhill in terms of an oxidation, reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain, but are passed on to the pigments of photosystem 1. Okay, so then what happened? The electrons in the reaction center, one excited and they receive red light at which wavelength, children? 700 nanometer, and a transfer to another acceptor molecule. Okay, so again these molecules move downhill. Then at the time a molecule energy rich NADP, first byproduct, is released. The addition of these electrons reduces NADP plus to NADPH and H plus. This whole scheme of transfer of electrons starting from the photosystem 2 uphill to the acceptor, down the electron transport chain to photosystem 1, excitation of electrons transferred to another acceptor, and finally downhill to NADP plus, reducing it to NADPH, and this is known as Z scheme. It is there in our textbook. Okay, same diagram is there in our textbook. It is called Z scheme. And during that time a molecule of water is split. H2O is split to 4H plus oxygen and 4 electrons. So this oxygen actually as a byproduct, this oxygen is liberated. Okay, now let's discuss the cyclic photophosphorylation and non-cyclic. In cyclic, what is happening? Living organism have the capability of extracting the energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. So the process through which the ATP is synthesized by cells is named as phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and IP, that is inorganic phosphate, in the presence of light. Okay, so light is required. When the two photosystems work in a series, first which photosystem will work? PS2 and then PS1. Okay, so this process is called non-cyclic photophosphorylation. The two photosystems, so here you can see the two photosystems work in a series. Okay, they are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH are synthesized by this kind of electron flow. Children, when PS1, that is photosystem 1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons. A possible location where this could be happening is in the stroma lamella. That is of the structure, you know, so at the stroma lamella it is happening, while the membrane or lamellae or the grana have PS photosystem 1 and 2, photosystem 1 and photosystem 2, the stroma lamella membrane lack photosystem 2 as well as this NADP reductase enzyme. So what happened? The excited electron does not pass on to NADP plus but is cycled back to photosystem 1 complex through the electron transport chain. Okay, so the cyclic flow hence result only in the synthesis of ATP. See, this is cyclic flow for the synthesis of ATP, whereas this one, both ATP and NADPH. So here only ATP is released. Okay, cyclic photophosphorylation also occurs when only light of waves beyond 680 nanometer are available. Clear, children? The difference is clear. Now let's discuss the chemiosmotic hypothesis. It has been put forward to explain the mechanism of ATP synthesis. And okay, so like in respiration and photosynthesis too, ATP synthesis is linked to development of a proton gradient across the membrane. So here you can see low H plus ion concentration and high H plus ion concentration. So how this proton gradient is formed? That we are going to explain. So there is one difference though. Here the proton accumulation is towards the inside of the membrane, that is in the lumen. In respiration the proton accumulate in the intermembrane space of the mitochondria when electrons move through the electron transport chain. Do you remember it, children? So now you just listen the steps. Since splitting of water molecules takes place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoids. Okay, as electrons move through the photosystems, what happens to the protons? Protons are transported across the membrane. So this happens because the primary accepted electron, which is located towards the outer side of the membrane, transfers this electron not to an electron carrier but to an H carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. So, the NADP reductase enzyme is located on the stroma side of the membrane along with electrons that come from acceptor of electron of photosystem 1. Protons are necessary for the reduction of NAD plus to NADPH plus H plus. So these protons are also removed from stroma. Okay, and you just observe this enzyme. This is ATP synthase. So ADP, by using inorganic phosphate in the enzyme, it produces ATP. Okay, so that is about the chemiosmotic hypothesis. Okay, so study well, children. Let's discuss the remaining in next class. Okay.